3. Mendelism. Subtopics: Monohybrid cross. It reveals how one trait is transmitted from generation to generation. Although inheritance of biological traits has been recognized for thousands of years, the first significant insights of how it takes place only occurred about 45 years ago. So Gregor Johann Mendel published results of experiments that would lay the foundation of formal genetics. So his works were kind of ignored until the turn of the 20th century, and then after his works were discovered, people established that genes are the distinct hereditary unit in genetics. Okay, when Mendel began studies using pigeon cerebrum or the garden pea, chromosomes were unknown, mechanism of analysis were also unknown, but he knew that, and also he determined there's a discrete unit of inheritance and predicted his behavior in the formation of gametes. And the investigators after that, now with cytological data, meaning that they are able to look at cells, they know that, oh, Mendel was correct all along. So all of the things that they found in cell, they follow Mendel's laws. Now that correlation of this thing were recognized, Mendel postulate was accepted as the basis of study of what is known as transmission genetics, how genes are transmitted from parents to offspring. So this principle was directly derived from Mendel's implementation. Even today, they serve as the cornerstone of a study of inheritance. So Mendel's model experimental approach to study patterns of inheritance. So he reported the results of genetic crosses between strain of garden pea in 1865. He is not the first to provide experimental evidence, but he succeeded where others have failed because of the experimental design and analysis. To actually do something that is good in terms of experimental biology, you actually need insight of how things work. Okay, to make your work as a researcher easier. So what he did, he uses an organism which was easy to grow and to hybridize artificially. The pea plant is self-fertilizing in nature, but you can also crossbreed it experimentally. Other than that, it has a very short life cycle. It grows and matures in a single season, and Mendel followed 7 visible features, so we refer to it as characteristics. And then he selected 6 other visibly contrasting pairs of traits involving seed shape and color, pod shape and color, flower color and position. All of these are quite clear to see, clear to the eye, meaning that when you see it, you can tell. From local seed merchants, Mendel obtained the true breeding strains, those in which each trait appeared unchanged generation after generation in self-fertilizing plants. Other than that, Mendel's success having other factors which make it successful. First of all, whenever he does experiment, he only studies 2 traits which are contrasting, not all of them, just 2, 1 versus 1. And then he also kept records. It is very necessary in genetic experiments. For analysis of his data, Mendel derived certain postulates that have become principles of transmission genetics. The Mendel's experiment went unappreciated until the turn of the century, well after his death. But once his studies have been rediscovered by geneticists in the modern times, he found out that Mendel was right all along. He had discovered the basis of transmission of hereditary traits. So, his similar crosses is just a monohybrid cross, one pair of contrasting traits. So you made a true breeding individual from 2 parent strains, each one of them have different or opposing traits. And then after we cross them together, we look at the first generation, and then the offspring of first generation, we cross them together to look at the second generation. So about the first generation, when we breed them all to each other, we call it self-fertilization or selfing. So the parents, they are the P, parental generation. Offspring number 1 in the first generation is called F1, and then after we self-fertilize them, we have the F2 or second generation. So the cross between 2 breeding pea plants, tall and dwarf, it is the representative of Mendel's monohybrid crosses. Because tall and dwarf, there are 2 contrasting traits of stem height. So unless tall or dwarf plants are crossed together with another strain, they undergo self-fertilization. So they will always break through. That if you just plant tall by itself, tall pollen will propagate and fertilize the tall flowers or stamen to become also tall seeds. Tall seeds will grow into tall trees. So they will always break through tall, 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 tall. Okay. So when Mendel crossed tall tree plants and dwarf plants, what he did is that he actually take the pollen from the tall and put it on the dwarf flower, and he did it vice versa. Take the pollen from the dwarf and put it in the tall flowers. Okay, so what happens that in both processes, the F1 generation are all tall. So when the members of F1 generation were selfed, Mendel observed that 787 out of 1064 F2 plants were tall, while 277 were dwarf. In this cross, you can see that in the F1 generation, there is no dwarf, but suddenly in the second generation, the dwarf trait reappears. So these were important in Mendel analysis of monohybrid crosses. So in genetic data, we express it as ratios. In many identical P crosses, all tall will produce, but in F2, if you try to use the numbers, okay, number of the trait of tall versus trait of dwarf, then the trait of yellow versus trait of green. Okay. When you do that, you might notice that all of the ratios are close to 3 to 1. Okay? No matter which trait that Mendel has crossed, they always turn up into 3 to 1, similar to the tall dwarf cross just described. And so all F1 offspring have the same trait exhibited by one parent. In the offspring, 3 to 1 is the ratio, meaning that of all of the plants, 3/4 follow a parent, while the other quarter follow the other parent. This other parent, the trait has disappeared in the F1 generation. Okay, so there's another further aspect of the monohybrid crosses. So in each cross, you have F1 and F2 patterns of inheritance were similar, irregardless of which gives the trait. So no matter if you have a tall sperm or dwarf egg, or you reverse it, dwarf sperm, tall egg, they will always happen. So in a nutshell, Mendel's monohybrid crosses were not sex dependent. So to explain these results, Mendel proposed the existence of particular unit of factors of each trait. So he suggested that these factors are the uniques of heredity, and they pass unchanged from generation to generation. Using these general ideas, Mendel proceeded to hypothesize precisely how such factors will account in the result of monohybrid crosses. So Mendel has 3 postulates. First postulate is unit factors in pairs, and then second postulate will be about the dominance and recessiveness, and third will be segregation. So unit factors in pairs. Genetic characters are controlled by unit factors in pairs in individual organisms. In monohybrid crosses involving tall and dwarf, a specific unit factor exists for each trait. Okay? So each diploid individual receives one factor from each parent, the mother or the father. Because factors occur in pairs, you can have 3 combinations: tall tall, tall dwarf, or dwarf dwarf. So there are 3 combinations. Each individual possesses one of these 3 combinations, which determines stem height. Dominance and recessiveness. 2 unlike unit factors responsible for a single character are present in single individual. One unit factor is dominant to the other, which is said to be recessive. In each monohybrid cross, the trait expressed in the F1 generation is controlled by the dominant unit factor. And so, the one trait that doesn't get expressed, we call it the recessive unit factor. And so, we can also put the term recessive and dominant on traits. So, whenever we can see that the traits, they only are found in second generation, they are usually recessive, because they do not turn up in the first generation. In the first generation is usually dominant, which is visible. So we can say that in Mendel's dwarf versus tall cross. Tall is dominant, and the dwarf is recessive. So segregation. During the formation of gametes, the pair unit factors separate or segregate randomly, so each gamete receives one or the other in equal likelihood. Okay, so each one of the gametes, they can either get a big T, small t, big T, small t. So big T, small t, they are unit factors. Each individual would have two unit factors. In gametes, the gamete only receive one, so half of it. Each gamete will have 50% probability of receiving either tall or dwarf unit factor. So there are three postulates that provide a suitable explanation for the result of monohybrid crosses. So Mendel reasoned that P1 tall plants have identical pair unit factors, just like the dwarf plants. So the gamete of tall plants receive one tall unit factor as a result of segregation, and the gametes of dwarf plants receive one dwarf unit factor, meaning that P dwarf and P tall, they will only give one T, big T, and one small t, one tall, one dwarf to each other. And then this offspring, each one of them will receive one unit factor for each parent. One tall for the tall parent, one dwarf for the dwarf parent. When F1 plants form gametes, segregation demands that each gamete randomly receive either the tall or dwarf unit factor. So following random fertilization events during F1 selfing, 4 F2 combination will result with equal frequency. So let's say that in F1 generation, they are big T, small t, right? Okay, so what if they combined with each other? What if they crossed with each other? So one parent will have big T, small t, the other parent will also have big T, small t. So we now have 4 different combinations: big T, small t, small t, big T, and small t, small t. So tall, 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 dwarf, dwarf, tall, or dwarf, dwarf. Combination 1 and 4 will result in tall and dwarf. But in dominance or recessiveness postulate, you have tall, dwarf, and dwarf, tall. You know that tall is recessive, is dominant. Then both 2 and 3 will yield tall plants. So in F2, we predict that we have 3 out of 4 tall and 1 out of 4 dwarf. So it is exactly what Mendel observed in the cross between tall and dwarf plants. So in other monohybrid crosses, this can also be found. So if you want to use modern genetic technology, we have to actually use Mendel theory, and then we have to explain it in modern terms. So in terms of a unit, Mendel calls it unit. Okay, each one of them will have unit factors, but we call it phenotype. Phenotype will be physical expression of a trait. Mendel's unit factors, it is a unit of inheritance. Modern geneticists call it genes, because genes are the sequence that is within your DNA that codes for a certain trait. Each one of the traits, such as plant height, dwarf, tall, yellow, green, all of these are controlled by genes. So geneticists also have several different systems for using symbols to represent genes. So if you want to refer to recessive trait, you use lowercase italic for allele for recessive trait. And for dominant, you use the uppercase italic. Okay, and then for pea plants, there's a lot of traits. Let's say that we want to use tall and dwarf, okay, D and T. But D and T are different letters, so we, according to the system, we actually have to use the letter of the recessive, meaning that we have to use D. So for tall, capital D, and for dwarf, small d. Yeah, what about the other traits? Okay, so for other traits such as green color and yellow color, usually yellow is the dominant compared to green. So what letter do we use? We use G. Big G for yellow, small d for green. Okay, so by reading the genotype, is written with the 2 letters, big D, small d, right? So this genotype is the genetic makeup of an individual for the trait. So by reading that genotype, we know what trait they will carry. Big D, two dominants, they will be tall. Weakly small d, one dominant, one recessive. The dominant always overpowers the recessive, so you have tall. Small d is dwarf. Okay, what about the sameness of the letters? So if you have a same letter, we call it homozygotes. Homozygous for the trait, or we call it homozygotes. But if the letters are different, big D small d, big D small t, big S small s, then we call it a heterozygote. So Mendel approach is analytical. So what led them to reduce the infected existing pairs? Because there are two pairs, because there were two contrasting traits for each one of the characters that he chose. So it's logical that two distinct factors must exist. So logically, that's what Mendel thought. You have tall and dwarf, just two types of plants. So there must be two distinct factors. However, why does one of the two traits of phenotype disappear in the F1 generation? If you look at F2 generation, we can answer the question. Because recessive trait do not actually disappear, it's just suppressed or hidden by the dominant. So usually, you have dominant and recessive together, the dominant will be overpowering the recessive. Therefore, Mendel concluded that one unit factor for tall and one for dwarf were transmitted to each one F1 individual, but the tall factor is dominant, so it has suppressed the dwarf factor. All F1 plants are tall. So given this information, we can ask how Mendel explained the 3:1 F2 ratio. As shown in figure 3.2, Mendel deduced that the tall and dwarf alleles of F1 heterozygote segregate randomly into gametes. If fertilization is random, this ratio is predicted. If a large population of offspring is generated, they will turn out with 3:1 ratio. So this man named Reginald C. Punnett, he devised the Punnett square approach. When you use a diagram called Punnett square, it is actually the easiest way to visualize the combining of gametes from different parents during fertilization. So in figure 3.3, you can see there is a F1 monohybrid cross, right? So this F1 cross has two heterozygous organisms, big D small d, big D small d, tall, right? So each one of these will create two gametes, a gamete for big D, gamete for small d. We make a Punnett square based on how many gametes are there. So for the first parent, there are two gametes. For the second parent, there are two gametes. So we make a 2x2 box, meaning that a box with 4 smaller boxes. Okay, so on top we put the gametes or the allele for the gametes on top for the first parent, big D, and then next will be small d. And then on the rows, you actually put the allele that the gametes hold. So big D and small d also. Then after you assign the gametes on their columns and rows, you predict the next generation by entering the male and female gametic information. So big D will enter the first box, and then big D will also enter the first box coming from the row. So you have big D, tall. The next, the small d will enter the box, and then from the row, the big D will enter the box. So you have big D, small d. So what happens that after you have done with the Punnett, you now have the number of the offspring that has a certain genotype. So in that box, now you have big D, big D, small d, big D, and small d. So what you have now is you can determine the genotype and the phenotype of this cross. So in the results, you have 1 big D, 2 big D, small d. So 3 out of 4 will be tall, but only one box would have a small d, which is dwarf. So phenotype will be 3:1, 3 tall, 1 dwarf. In terms of genotype, you have 3 genotypes: 1 big D, 2 big D, small d, and 1 small d. So in terms of genotype, the ratio is 1:1. The Punnett square enables us to list all possible random fertilization events, and from there we can know that what kind of genotypes and phenotype will come out, and it is very useful when you're first learning genetics and how to solve genetic problems. So you look at this exercise, okay, 3.1, which is dominant, which is recessive. which symbols are assigned, and what are the genotypes of the parent. So, test cross, one character. So test cross is done when you want to determine the genotype of an unknown dominant organism. So so let's see that the top plants can either be big D small d or big D big D. So you don't know what. So how do you distinguish between them? Physically, they are undistinguishable, but Mendel has devised a rather simple way to determine what kind of genotype that they have. So they have to go under test cross. So the organism that has the dominant phenotype will be crossed with a recessive phenotype. Means that to know what kind of genes or what kind of allele that the mysterious dominant one has, you have to cross it with a recessive. So you have big D. Okay, if you cross it with small d, you can be either all tall or half tall, half dwarf. If the test cross would have all tall, meaning that the tall organism, the tall tree, the tall pea plant only has big D alleles. It doesn't have any other. But if the test cross results in half tall, half dwarf, meaning that tall plants actually are heterozygous. It has the dwarf genes. The Mendelism cross generated a unique F2 ratio. So to extend the Monohybrid cross, Mendel also designed an experiment in which he examined two characters simultaneously. So this cross is called the dihybrid cross or two-factor cross. So if we have pea plants having yellow seeds that are round, compared with having green seeds which are wrinkled, so means that you now have two traits, the color and also the shape of the seeds. So figure 3.5 would occur. You have F1 offspring, all yellow, all round. Okay, meaning that yellow and round are the dominant traits. Yellow is dominant to green, round is dominant to wrinkle. When the F1 individuals are selfed, now you have very unique ratio 9:3:1. 9 will be all dominant, 9 yellow and round, and then you have 3 which are dominant in one trait and recessive in the other. So you have 3 with yellow and wrinkled. In that in color they are dominant, in shape they are recessive. And you also have another 3 which are also dominant in one trait but recessive in the other. But in this case, the traits which are recessive and dominant are swapped. I mean that you can see green round, that they are recessive in the color but they are dominant in the shape. And 1 out of 16 will be all recessive, green and wrinkled. So in this variation, we have one pea pattern with dominant to one with recessive. So we have primary yellow wrinkled seeds are crossed with the green round seeds. So we swap the actual traits around. So we have now one dominant allele, but also one recessive allele. Meaning that these parents would have one recessive and one dominant trait. But still, the result would remain unchanged. Okay, now for the Mendel's fourth postulate. The Mendel's fourth postulate, you have the dihybrid cross. Okay, so dihybrid cross, you have two traits, right? Two traits, they are working or interacting with each other. But the thing is, these traits are not actually resulting in influencing the other trait. Meaning that the green allele does not influence the wrinkle allele. So we can say that the traits are independent of each other. So this postulate was created from the dihybrid cross. Okay, so Mendel examined the F1 plus of dihybrid cross, all yellow, all round. This is our theory. The result will be 3 to 4 yellow and 1 for green. Second cross, 3 to 4 round and 1 to 4 wrinkle. Meaning that if you look at trait by itself, it will always follow 3 to 1 ratio. Okay? meaning that it doesn't actually follow the other traits. So you have the trait for color, and then you have the trait for wrinkle. So if you just look at this diagram, okay, you have yellow round, okay, you have yellow and green. Okay, somehow one pen is not working. Okay, so you can see that you have okay, let's look at this color, yellow and green. Okay, they cross, and then what you get? You get yellow. Okay, and then when you do it again, F1, how many yellow do you get? Okay, you get 9 plus 3 is what? 12. And then how many green you get? 3 and 1, 4. So right, so 12 and 4, what is the ratio? 3 to 1 also, meaning that if you look at each trait by itself, it is 3 to 1. So this number demonstrates that the two pairs of contrasting traits are inherited independently. So we can predict the frequencies of all possible F2 result by applying the product law of probabilities. What is product law? Product law means that there are two events that happen simultaneously. So each one of the event does not influence the other events. So if one look at the probabilities, they are equal to the product of the individual probabilities of occurrence. For example, flipping two coins mean that you flip two coins, a coin will have head and tail. So what if we want to look at the probabilities of having two tails? To get tail, the probability is 1 over 2. To get the next tail is also 1 over 2. So to get two tails, it's 1 over 2 multiplied by 1 over 2, you get 1 over 4. Means that in flipping two coins, you can get either head, two heads, or tail, head, or maybe head, tail. And then to get a tail, it is 1 out of 4. For example, the probability of an F2 plant having yellow and round seeds is 3 out of 4. Okay, 3 out of 4, or 9 over 16. Now 3 out of 4 is of all plants should be yellow. 3 out of 4 of all plants should be round. Means that you have two traits. The trait for yellow and then trait for the shape. Okay, so the trait to actually get the yellow color is 3 out of 4. That's the probability. And the probability to actually get the round, which is the dominant shape, is also 3 out of 4. So let's say that, okay, what if we want to get yellow and round together? To actually get yellow and round together, we just multiply them. 3 over 4 multiply with 3 over 4. So we get 9 over 16. So you can also calculate the other probabilities. We say that you want to ask, okay, what is the probability of getting a green and a round? So green is what? 1 out of 4. To get round is 3 out of 4. So 1 out of 4 multiplied by 3 out of 4, you get 3 out of 4. 3 out of 16, sorry, not 3 out of 4. 3 out of 16. 3 out of 16 will be green and round. F1 and F2 results are identical whether the initial cross is yellow, round plant by green, wrinkle plants, or whether yellow, wrinkle plant by green or plants. In both crosses, F1 and F2 are identical, and as a result, F generations are also identical in both crosses. Based on the similar results of numerous hybrid crosses, Mendel proposed a fourth postulate, which is independent assortment. So for independent assortments, during gamete formation, circulating pairs of univectors assort independently of each other. This postulate stipulates that segregation of any pair of univectors occurs independently of all others. As a result of random segregation, each gamete receives one member of every pair of univectors. So let's say that we have A, B. A is a trait for trait 1. Let's say that it is for tallness. B is another trait, maybe a trait for skin color or something, right? So each one of the gamete must have a member of A and B, meaning that you can get either big A, big B, or maybe you can get big A, small b, or maybe you can get small a, small b, maybe you can get small a, big B. As long as you have a member of A and B, A can go into any cell independently from B. B can go to any cell independent from A, because usually they are in other chromosomes. So the Bonnet square in figure 3.7 shows how independent assortment works in the formation of F2 generation. So when you examine the formation of gametes by F1 plants, the aggregation to prescribe that every gamete will have either big G, or big W or small w. Okay. Independent assortment stipulates that all four combinations will be formed in equal probabilities. Okay. So when you want to write the Punnett square, you must take notes of what kind of gametes are there, and you must write all combinations. Okay. So you have big G, big W, big W, small g, small g, small w, small w. So you have big G and big W, right, to make F1 yellow. So if F1 yellow round, you can see it is big G, small g, big W, small w. So we write all the combinations: big G, big W, big G, small w, small g, big W, and small g, small w. So when you want to do Punnett, you have now 4 gametes, and the other parent will also have 4 gametes. So how big is your Punnett? 4 by 4 is 16 boxes. If mendel springs are produced, 9 out of 16 will have yellow round, meaning that dominant. 3 out of 16 will have yellow wrinkle, meaning that dominant recessive. 3 out of 16 have green and round seeds, recessive dominant. And 1 out of 16 will have green wrinkle, recessive. So this is the ideal ratio, 9-3-3-1 dihybrid. What if we have 2 dominant traits, but we don't know whether it is heterozygous or homozygous? Same thing with the monohybrid, we do a test cross. So test cross with a plant or an organism with a double recessive. Okay, let's see that we have tall and green. Okay, we cross that organism with a dwarf and yellow. Okay, let's see what happens. So if an F2 yellow round plant is crossed with the homozygous recessive green, you will get the offspring, which are, there are 4 results that might happen. So if the yellow round has a big Y, big R, and then when it meets the gamete, which is small y, small r, which is the double recessive, you get yellow round. If you have a gamete which is big Y but small r, in that in the color they will be dominant, yellow, but in the shape they will be wrinkled. Okay. Other than that, if you have small y, big R, when it meets the double recessive, you have green round. Okay, small y, small y, big R, small r, right? Then if you have the small y, small r, what happens is that you will have green wrinkled, in that both traits will be recessive. To be continued.